has some clefts or active sites. So active sites are needed for the enzymatic activity. Hence, the proteins that exhibit tertiary structure can only act as enzymes. All the enzymes are proteins, I said, but there is an exception. What is that? Ribozyme. So ribozyme is an enzyme, but it is not a protein. So that is a RNA. So nucleic acid, ribonucleic acid. So a RNA acts as enzyme, that is a catalytic RNA. Such one is called ribozyme. Ribozyme is an enzyme but not a protein. That is an exception to that statement that all enzymes are proteins. This is not protein. This is RNA, catalytic RNA, whose sedimentation coefficient is 23S. A type of rRNA acts as enzyme, that is called ribozyme. Ribozyme, we know very well, ribozyme is responsible for the formation of peptide bonds among the amino acids at the time of protein synthesis. And the first discovered enzyme is gymase, and the first crystallized enzyme is urease. Urease was crystallized by the James Sumner from the Canovalis elsiformis or jog bean, whereas the enzyme gymase was discovered by Edward Buchner from the East. The study of the enzymes is generally said to be enzymology. Okay, coming to the how they carry out the reactions. What is the function of enzyme? Why enzymes are used? And what is the difference between inorganic catalyst and organic catalyst? This is organic catalyst, biocatalyst, otherwise organic catalyst, because chemically it is made up of protein. The difference is inorganic catalysts can tolerate high temperatures and pressures, whereas the organic catalyst can't tolerate the high temperature. Above a limit of temperature, after a certain temperature, the enzymes undergo chemical destruction through the process known as denaturation. Of course, some exceptions are there even in the biocatalyst also, like Taq polymerase. Taq polymerase is the enzyme which is used in the PCR technique, you might have known. So that was extracted from the Thermus aquaticus bacterium, so which even function above 90 degrees. That is an exception. But in general, after 45 degrees like that, enzymes, they undergo denaturation. They undergo chemical destruction at high temperatures through the process known as denaturation. Whereas the catalyst, they are thermotolerable. They can work out even at high temperatures and high pressures. That is the difference between the inorganic catalyst and organic catalyst, like biocatalyst or enzyme. Okay, so what are the types of reactions? Here, two types of processes are there, like chemical processes and physical processes. For instance, if you take the ice, ice, it is melted into water. Water, if you overheat, it gets vaporized. So ice is converted into water, water is converted into vapor. Is it a chemical reaction? No, we cannot consider it as a chemical reaction because it doesn't involve either making or breaking of the bonds. Just this is change in the state of matter. A solid is converted into liquid and a liquid is converted into vapor, gas. That we cannot consider as chemical process. That is a physical process. Coming to the chemical processes, what are they? Either it involves in the making or breaking of the bonds. If either making or breaking of the bonds takes place, that we can consider as chemical processes like this. You see, barium hydroxide reacts with H2SO4, resulting in the formation of barium sulfate and water. So here, making of the bonds occur and breaking of the bonds also occur. So bonds are broken, bonds are made. So as it involves the making and breaking of the bonds, this is said to be chemical process or chemical reaction. But is it organic reaction or inorganic reaction is the question. So this is undoubtedly this is inorganic itself. How? Because no carbon. This is, for instance, you take this, starch is broken down to glucose, starch is hydrolyzed to glucose. Even this is also a type of chemical reaction. So this is organic chemical reaction, whereas the above mentioned is inorganic chemical reaction. When starch is broken down to hydrolyzed, then glucose are formed. Many glucoses are the monomeric units which make up starch, and hydrolysis starch gives rise to again glucose itself. So this is the organic chemical reaction, whereas the above mentioned one is inorganic chemical reaction, and this representation is a physical process which brings only change in the state of matter without involving the either making or breaking the bonds. So rate of reaction, the formula for rate of reaction is shown here. The amount of product produced per unit time is said to be rate of process or rate of reaction. Coming to the types of reactions, catalyzed, uncatalyzed reactions. What is catalyzed reaction? The reaction that takes place in the presence of enzyme is said to be catalyzed reaction. If the reaction takes place in the absence of enzyme, it is said to be uncatalyzed reaction. For instance, you take this carbon dioxide reacting with water molecule resulting in the formation of carbonic acid. This reaction may take place in the presence of enzyme or even absence of enzyme also. You suppose if it takes place in the absence of enzyme, only near about 600 molecules, 600 products, 600 molecules of the carbonic acid are 600 molecules only formed per hour. For 1 hour, 600 molecules are formed. Same reaction if it takes place in the presence of enzyme. What is that? Carbonic anhydrase. If this reaction takes place in the presence of carbonic anhydrase enzyme, that here, it is near about 200, whereas if it takes place in the presence of enzyme molecule, 6 lakhs products are produced per second, not per hour. So what does it mean? So here the rate of reaction is dramatically increased because it is taking place in the presence of enzyme. What is that? Carbonic anhydrase. Like this, catalyzed reactions are very rapid as compared to the uncatalyzed reactions. Very clearly seen here. If it is uncatalyzed, only 200 molecules are formed per hour, whereas if it is catalyzed, 6 lakhs molecules are formed per second. So like that, catalyzed reactions are more rapid as compared to the uncatalyzed reactions. So reactions, they may take place singly or they may take place in a series. If they take place in a series, that series of reactions is said to be metabolic pathway. Like this, glucose is converted to pyruvic acid. So this is nothing but glycolysis, fundamental respiration or common respiration, which is part of both aerobic respiration and anaerobic respiration. In glycolysis, glucose is converted to pyruvic acid in general, and the end product varies from organism to organism or depending on the conditions and situations. For example, in the absence of the oxygen in the skeletal muscles, lactic acid is produced in the same series of reactions. In the presence, pyruvic acid is produced. If it is yeast, alcohol or ethanol is produced. So like that, end product varies depending on the situations. However, in the same metabolic pathway, different products are formed. Okay, how the enzymes work out? Actually, enzyme is a macromolecule to which substrate gets attached so that enzyme substrate complex is formed. Enzyme substrate complex, C. Later, enzyme substrate complex is converted to enzyme product complex. Finally, substrate is converted to product. Enzyme will be released as it is. Enzymes are not used in the chemical reactions, just the speed of the chemical reactions. At the ground state, we cannot make or break bonds of this substrate molecule. So, we should bring this substrate molecule to its a transition state. In the transition state, we can make or break the bonds in this substrate molecule. So in a chemical reaction, first substrate molecule is attached to enzyme, later substrate molecule is reached to its transition state, one or two states, many or any number of states may be there. Through that structural transition states, it undergo finally there at the transition state, either making or breaking of the bonds takes place, resulting in the formation of product. Here, to bring this substrate molecule to its transition state, we need to invest some energy. That energy is said to be activation energy. Activation energy is the energy required for the occurrence of a chemical reaction. Otherwise, activation energy is a difference between the potential energy content of the substrate molecule and its transition state. To reach the transition state, it needs some energy. For example, here 100 kilocalories potential energy is present in the substrate molecule, but its transition stage or state, it is 150 kilocalories because there is energy input to activate the substrate molecule to reach the transition state. So the difference between this is 100, here totally 150, here 50 we added, it become 150 means 150 minus 100, 150 minus 100, that becomes 50. So that is activation energy which is needed to activate this substrate molecule, or to bring the substrate molecule to its transition state, we have to input 50 kilocalories of energy. So how can we define activation energy? Activation energy is the energy, the difference in the energy levels of the potential energy content of the substrate molecule and that of its transition state. Okay, if you see, this activation energy, it fluctuates depending on the presence or absence of the enzyme molecule. You suppose, if this reaction is taking place in the presence of an enzyme, activation energy is very less. So, with a little amount of activation energy, substrate molecule is converted into product. When? In the presence of enzyme. When you use enzyme, activation energy will be lowered. So that is a function of enzyme. Enzymes speed up the chemical reactions by lowering the activation energy. You see this? So this curve you see? Without enzyme, activation energy, more activation energy is needed without enzyme. With enzyme, less activation energy is needed. That is the function of enzyme. It lowers the activation energy. By lowering the activation energy, it speeds up the chemical reactions. And you see, you suppose here, the potential energy content of the product is more as compared to substrate molecule. When it is more, if it is endothermic reaction, if the potential energy content of the product is more than that of substrate molecule, such reaction is said to be endothermic reaction. Endothermic reaction means it takes the energy. There is energy input in the reaction. That is why potential energy content of the product is more than that of the substrate molecule. Endothermic reaction. Or you can say that it is a anabolic process, or you can say that it is a endorganic process, or simply you can say that it is a constructive process. Constructive process. For instance, photosynthesis. Photosynthesis is a constructive process. There, the energy level or the potential energy content of the glucose is more than that of carbon dioxide and water. So that is I said here. If the energy content of the product is more than that of substrate molecule, such reaction is said to be endothermic reaction, or anabolic reaction, or endorganic reaction, or constructive reaction. Like that, if it is less, if the potential energy content of the product is less than the substrate molecule, then that is called exothermic reaction. See, exothermic reaction because energy is released, or that is called catabolic reaction, or the same is called exergonic reaction, and it is called destructive reaction. Exothermic, catabolic, exergonic, and destructive. Simple, two terms. If the potential energy content of the product molecule is less than the substrate molecule, such reaction is said to be exothermic. If it is more than the substrate molecule, that reaction is said to be endothermic reaction. So this x-axis represents the progression of the reaction, whereas the y-axis represents the potential energy. Okay, coming to the mode of enzyme action. Here, enzyme molecule, this is substrate molecule. Enzyme is a macromolecule, substrate is micromolecule. Substrate is attached to enzyme molecule at active site. Active site is a place of enzyme where substrate molecule gets attached to it. When substrate is attached to enzyme molecule, enzyme substrate complex is formed. E, enzyme, S substrate, C complex. Means substrate, now it undergoes some transitional changes. It reaches its transition state. At that state, either the expected making or breaking of the bonds takes place, and substrate is converted to product, so that the enzyme product complex is resulted. Finally, enzyme is released as it is, and substrate is converted into product. Actually, this is the representation of the enzymatic activity. To explain the enzymatic activity, different models are proposed by different workers. Of them, the popular ones we discuss here. See, lock and key theory. Lock and key theory was given by Emil Fischer. According to the lock and key theory, the configuration of the substrate molecule and that of active site should be similar. Then only reaction takes place. Otherwise, reaction will be inhibited. Reaction will not take place. Like a key and keyhole. When the configuration of key and keyhole is similar, then only the key can be inserted and the lock can be unlocked. With the same key, we cannot unlock all the locks. To unlock a lock, the configuration of the key and keyhole must be similar. So like that here also, when the substrate molecule configuration and active site configuration both are similar, then only enzyme substrate complex is formed and the substrate is broken down to product and enzyme will be released. Then only reaction takes place. According to this theory, here active sites are very rigid. They do not undergo any conformational changes. If the same type of substrate molecule which resembles the configuration of the active site comes, then only reaction takes place. Otherwise, reaction is stopped. If suppose here, this is enzyme molecule, active site. Substrate molecule configuration is similar, so it can fix into this. So ES formed, E and P also formed. In this case, you see, configuration of the substrate molecule is not similar to that of active site. So this substrate molecule cannot get attached to the active site because the configuration of the substrate molecule and active site are dissimilar. In this case, reaction doesn't take place. Coming to the one another proposed by Coastland, and the theory is said to be induced fit theory. According to the induced fit theory, always reactions takes place. In no case reaction is stopped depending on the configuration of the substrate molecule, because here you see this first case, active site configuration, this is substrate molecule configuration, both are similar, it can be fixed, ES is formed, EP is formed, product is formed, enzyme released. even though it is not similar to the active site. Actually, active site is like this, but the substrate molecule is like this. The configuration of the substrate molecule is not similar to that of active site. It doesn't resemble with this. Even though reaction takes place, how this substrate molecule, it induces some conformational changes in the active site of the enzyme molecule. Depending on the configuration of the substrate molecule, active site of the enzyme molecule undergoes some conformational changes to get a configuration which is similar to the substrate molecule. So, whatever it may be the shape of the substrate molecule or configuration of the substrate molecule. Depending on the configuration of the substrate molecule, active site undergoes some conformational changes to get a active site with configuration similar to the substrate molecule. So like this, here it induces some conformational changes in the active site in such a way that enzyme molecule gets an active site whose configuration is similar to that of substrate molecule. So no problem. Now the substrate molecule can get attaches into the active site, so that results in the formation of enzyme substrate complex, and finally substrate is converted to product and enzyme will be released as it is. So this is the induced fit theory, according to which the active sites are flexible. They may undergo conformational changes induced by the substrate molecule. So depending on the substrate molecule configuration, here active site undergoes some conformational changes to get a active site which is similar to the substrate molecule.